Hello everyone, welcome to my presentation about the future of personalized diagnosis and treatment for brain diseases within the Symposium about exploring sex and gender issues in neuroimaging research, insights from science and society, organized by Hyun Woon Lee and Randy Golub. I would like to thank the organizers for having me here. Unfortunately, I'm not able to be there in person, but I would be very happy to receive your feedback via email after the symposium. The studies that I'm presenting all use the open source virtual brain framework. The software is open source. As I mentioned, you can go to the website, virtualbrain.org, and just download it and use it for your purposes. The novelty of this approach is that we combine mathematical modeling with personal data. So we construct brain network models, usually based on individual neuroimaging data. Here you see how we extract the individual modalities or ingredients for a brain network model. We reconstruct, for example, the fibers using diffusion tensor imaging. We parcellate the brain using different parcellation methods with a different degree of granularity, kind of very fine-grained or functional parcellations or more coarse-grained parcellations. And then each brain area is equipped with a so-called neural mass model or mean field model. And then we use the structural connectomes to compute the result of the complex interactions, the flow of information amongst the different brain regions. And then we can simulate neuronal activity depending on the mean field model and the forward models that we are using, for example, fMRI build activity on cortical and subcortical structures, or with forward models we also can simulate EEG or MEG signals. And we even can use these brain models to do interventions, like surgical interventions, or just introducing artificial electrodes and stimulating the brain and simulating the dynamic effects of this stimulation. In the next few slides, and before I then switch to the pathway towards clinical application, I want to show you how we can use these brain network models to better understand mechanisms. So we want to understand how entities, for example, neuronal units, and activities, for example, information flow, interaction between the neuronal units are organized in such a way that they are responsible for the observations or phenomena that we record with our measurement methods, like fMRI, EEG, MEG, PET, and so forth. So for this, I want to show you three examples of three relatively recent publications of our groups where we used this brain network modeling approach to understand better principles of brain dynamics and also cognitive function. And the first study that I am using here as a demonstrator has been published in 2018 in eLife, with the title Inferring Multiscale Neural Mechanisms with Brain Network Modeling. There we have used the reduced Wang model. Here you see the equations and constructed individualized brain network models of 15 healthy adults. The multimodal imaging data we had acquired in Berlin, it's fMRI, EEG, diffusion tensor imaging data, and anatomical data. And with our simulations of 22 minutes of fMRI bold activity, we were able to, at the same time, infer for each simulated brain region the activity of the neurons, the firing rates, as you can see here. So here we see the trace of the excitatory populations, and here you see the trace of the firing rate of the inhibitory populations. So here if we see the equations, basically the input currents of the excitatory and inhibitory population are translated into firing rates of the excitatory and inhibitory populations that you can see here, and these are translated into synaptic activities. So we have all this information incorporated in our model when we are simulating the fMRI bolt signal that we have measured for those individuals. So this is an overview. I will go through the individual panels step by step, but this shows you in the upper panel the simulated results and in the lower panel the corresponding experimental data. And we have sorted this according to the time scale from 100 milliseconds time window to 22 minutes time window. And you can see that we are able to capture with our model many different features of experimental data that in experimental data can be assessed only with different methods and even only in different species, because some of the methods are invasive. So for example, here in panel A, we see in our simulation the relationship between the firing rates and the phase of the alpha rhythm. And we see here that at the peak of the alpha rhythm, we have a trough of our firing rates, and we have similar relationships in monkeys. In panel B, we see the amplitude relationship of inhibitory postsynaptic currents and excitatory postsynaptic currents in our simulations, and the corresponding data from rats. We see the firing rate versus the alpha power. Here we had the phase. Now it's a slower time scale. We look at the power. And we see in our simulation the inverse relationship between the firing rate and the alpha power, which has been also shown in monkeys experimentally. We here in panel D see the relationship between the alpha power and the bolt signal in our simulation, an inverse relationship, and the same has been shown in many simultaneous EEG-fMRI studies, also from our lab before. We see in our simulation the power law behavior of simulated fMRI data, which has been shown also experimentally in humans. And here, finally, we see in our simulations the switching of functional connectivity over time, functional connectivity dynamics, and this also has been shown experimentally. So with a single simulation, with a single brain network model, it is possible to capture many different types of observations or snippets of observations and to integrate them in one consistent unifying model, and thus to understand how these different observations mechanistically are mutually related. So this is a summary slide for the first study that I was going to present you to show you how we can understand principles or mechanisms of brain function. So here we see all the different, or some of the different components. We see the alpha rhythm modulations. We see how this leads to pulsed inhibition of the firing rate and to the modulation of the feedback inhibition signal. How this translates in slow fluctuations of the fMRI signal. Here you see the network effects, and here you see this inverse relationship between the alpha power and the bolt signal. And this is all encapsulated in a single brain network model and can be observed in these simulations with a single parameter setting. So we don't need to modify the parameters. Now I come to the next study that I want to demonstrate you to show you how we can understand mechanisms of brain function and dynamics. It has been published this year in Nature Communications as the title Human Connectome Topology Directs Cortical Traveling Waves and Shapes Frequency Gradients. We would now expect, given also from the previous experiments, that there is a direction of traveling waves from temporal parietal regions to frontal and occipital regions. This was tested with a simple model first, a coupled Kuramoto network model. We also considered the time delays of the structural connectome, and this is a simulation. And as you can see, we have such Kuramoto oscillators coupled with the structural connectome of the Human Connectome Project subjects. We see immediately these traveling waves, and of course the traveling waves also can be characterized statistically. Here you see, for example, the flow fields, the sinks, and the sources, and we see the red nodes that indicate the sources are distributed around the parietal temporal areas, and the sinks, the blue nodes, can be found in frontal and occipital areas. So the traveling waves travel from mainly from temporoparietal to frontal and to occipital areas. This publication published last year in Nature Communication with the title Learning How Network Structure Shapes Decision-Making for Bio-Inspired Computing. And here we were interested not only in the dynamics of the activity that is generated by our brain network models, but also how this relates to cognitive performance. And to this end, we again used the Human Connectome project data. We virtualized 650 brains, created 650 virtual brains of the adult healthy subjects. And we related our simulations to the cognitive scores that have been also obtained for these subjects, and in particular also to their fluid intelligence. We here in this study showed how functional connectivity relates in the model to the input amplitude that drives the node. There's a direct relationship, and this modulates again in this plugged-in functional circuit the ratio of correct decisions, the time until a decision is being made. And this can be completely explained by the state space diagram, where we see how the synchronicity in the network reconfigures the state space and leads, therefore, to the different performances. We also have developed the possibility for multi-scale co-simulation, so coupling the mean field network simulator virtual brain with other simulators like NEST and Anarchy, and to be able to run some simulations in some regions of the brain at a finer resolution, for example, spiking neuron networks. This has been started in this work led by Jill Meyer, where we used the multi-scale brain simulation possibility provided by the virtual brain to construct multi-scale brain network models of patients with Parkinson's disease, where the basal ganglia that are typically stimulated in these patients are simulated at a finer granularity.
Excuse me, can you stop the video?
Neurons.
Thank you.
With this, we now...
I'm apologizing to you. We need to stop because everybody has to rush over and hear the next keynote lecture because it's Nicola's talk and we can't miss it. So, in support of our women faculty who are awesome and who are giving great keynotes, I'm sending you all to the lecture.